So let us continue glycolysis that we discussed last Tuesday. So we stopped at the step after the hexose is cleaved into two trioses, and then we saw dihydroxyacetone phosphate isomerizing into glyceraldehyde 3-phosphate. So today we will continue from glyceraldehyde 3-phosphate, but at the end we will have an overview of all the 10 steps that form the glycolysis. So oxidation of glyceraldehyde 3-phosphate is essentially this aldehyde group, shown here on the left top, is getting oxidized into a carboxylic acid group, and the energy is conserved in making a phosphate mixed anhydride bond with an inorganic phosphate. That's one. And second part of the oxidation energy in terms of the electron transfer is used to reduce NAD to produce NADH. Okay, this carries the hydride version of electron transfer. So 2 electrons and protons, so 1 proton goes into the medium. So this is the first step of energy yielding phase of glycolysis. The steps we saw the previous ones, like glucose to glucose 6-phosphate, and then fructose 6-phosphate to fructose 1,6-phosphate. So those are preparative steps where the bonds are energized, like the poor leaving groups are replaced with good leaving group, in this case phosphate. So those steps are called the preparative phase. So now this is the first step where you see energy yielding. So energy yielding in the sense, here the electrons are transferred to reduce NAD. So that is one. And this NADH, if it enters into electron transport chain in mitochondria, will approximately yield 3 electrons per NADH, and here you have 2 glyceraldehyde 3-phosphate coming from a single glucose molecule. So as a result, you will get 2 NADH and therefore 6 ATP. So that possibility exists. Okay, so temporarily it is conserved here. And so let us look at its reaction mechanism. So this involves, so this is the active side of the enzyme, where you have the oxidized NAD bound, and then you have this non-covalent interaction between the sulfhydryls hydrogen and this electron-rich nitrogen, forming an interaction here. So this is the active site structure. So the first step is this chemiacetal, but it is a thiochemiacetal. So this is an alcohol group. This is an aldehyde. Remember, we learned about how a ring structure of glucose forms. The aldehyde of the hexose forms an intramolecular hemiacetal with one of the alcohol groups, which is the OH groups available in glucose molecule. Very similarly, here the aldehyde of this carbohydrate triose, instead of an intramolecular, like this would be an intramolecular alcohol group, this OH here, instead it is with an intermolecular and instead of alcohol, this is thioalcohol, forms a thiohemiacetal therefore. Okay, so again you can see this carbonyl group, this carbon will have partial positive charge, and as a result it is susceptible for nucleophilic substitution reactions, and that's how you get this thiohemiacetal, and that again undergoes oxidation to thioester form. So this is a thioester. This is no longer an alcohol group. Instead, it is an acyl group, and that is facilitated by the transfer of electrons from this to this NAD plus, and that's how NAD gets reduced, and this transfer leads to the formation of this ester bond, this carbonyl group. And subsequently, the reduced NADH is replaced by an NAD, and so the original state returns. And a phosphorylitic cleavage, instead of hydrolytic, where it is water, here it is inorganic phosphate. And so there again, this partial positive charge in this carbonyl makes that readily possible. And that is also aided by this general base catalysis possible by this imidazole nitrogen of the histidine chain. And that releases the product, which is 1,3-phosphoglycerate. So essentially, this thiohemiacetal oxidized to thioester gets phosphorylized, instead of hydrolysis. So it is exchanged with this. So you have a mixed anhydride, carboxylic acid, and then the inorganic phosphate. So you end up getting 2 phosphates. Okay. So this bond, conserves the energy made available by the oxidation of the original aldehyde group to the final carboxylic acid group. Okay, so this is a high energy phosphate, the one shown here, this carbon phosphate bond. So this is a 1,3-phosphoglycerate is formed. So here you have net gain of 2 electrons per glyceraldehyde, so 4 electrons per glucose conserved in the form of reduced NADH. And in the next step, which is the enzyme name is for the reverse reaction, 3-phosphoglycerate gets phosphorylated to 1,3-phosphoglycerate. So the enzyme name comes from that phosphoglycerate kinase. But in glycolysis sequence, this 1,3-phosphoglycerate, that one of the high energy phosphate bond, that is transferred by making an ATP molecule. Okay, so here ADP gets phosphorylated, forming the ATP, and the high energy bond here is now transferred to the formation of ATP. So this kind of a generation of an ATP molecule is usually called substrate level phosphorylation, meaning from a substrate of this enzyme, you have the phosphate group transferred to make ATP. This is in contrast to the oxidative phosphorylation based ATP formation in mitochondria, where the electron transfer from one electron donor to another electron acceptor leads to the formation of a proton gradient, and then that proton gradient is used to make ATP. So that is called oxidative phosphorylation. To distinguish from that, we call these kind of ATP formations as substrate level phosphorylation. So this is the first substrate level phosphorylation you are encountering in glycolysis. So there is one more step later where we will see substrate level phosphorylation happening. So here, this is the first step in glycolysis where you see ATP formation. So here, for one of the trioses, you end up making 1 ATP. So per glucose, you have generated 2 ATPs. So this balances the 2 ATPs used in making glucose 6-phosphate and then the fructose 1,6-phosphate. So right now, in terms of ATP, net gain is 0. The only thing is 4 electrons we abstracted have gone into making NADH. So NAD plus has been reduced. So that's the one gain. And the next step is simply an isomerization where the this ester is rearranged to the second carbon by mutase, phosphoglycerate mutase, that converts 3-phosphoglycerate to 2-phosphoglycerate, which is very important for the progression of glycolysis, because the next step involves a reaction that will generate another energy rich molecule. So this at the enzyme active site happens in this manner. The first thing is actually a phosphoester bond with the second carbon with an enzyme that is already phosphorylated. This histidine imidazole side chain is phosphorylated. That donates the phosphate group, making the 2,3-phosphoglycerate. And from this, you have the original, the third carbon phosphate transferred to the enzyme, so regenerating the enzyme's active site. So this is an important intermediate, a small quantity, but it's crucial to make the reaction continuously proceed. So this 2,3-BPG is an important molecule in the glycolytic regulation. So this is how the mutase makes the 2-phosphoglycerate. And the next step, so this is a 2-phosphoglycerate, and this is redrawn from this to make it easy for us to understand how the dehydration happens, leading to the formation of an enol structure. So this is catalyzed by enolase. This forms phosphoenol pyruvate. And this is a high energy compound. When we were talking about how ATP is a high energy compound, there we looked at a few other molecules, and one of them, if you remember, is phosphoenol pyruvate. Because hydrolysis of this ester bond leads to a alcohol group, so it will be an this will be an alcohol and this will be enol. Therefore, this double bond and the OH together we call enol, and it can undergo tautomerization to form keto-enol tautomerization. So that is sort of resonance kind of stabilization. Therefore, the hydrolyzed structure, which will be pyruvate, is more stable. So therefore, this we call as a high energy compound. And enolase generates this by dehydrogenation of the 3-phosphoglycerate. So the last step is that hydrolysis of the high energy bond. So you have another substrate level phosphorylation producing ATP molecules. So these two ATP, so far we have cancelled whatever generated and whatever consumed, and these two ATPs, like 1 ATP per phosphoenol pyruvate and 2 phosphoenol pyruvate per glucose. In that sense, per glucose 2 ATP. So this is the net gain from glycolysis apart from the 2 NADH produced. In terms of the ATP production, 2 ATP molecules are produced per glucose in glycolysis. So this structure produced pyruvate can be in keto-enol tautomerization, and due to that, that is more stable. So that is shown here. So this is the pyruvate in the enol form, and this is the keto form. Okay. So this is the carbonyl group. We call this the keto group, and this is the enol. So because this is an ene, alkane, alkene. So that ene, double bond. And here you have the alcohol group. That's where this all comes from. So that's why this is called enol. And this kind of an acid is called alpha-keto acid. In the old convention, this is not named, and this is alpha, beta, and so on. As for IUPAC, this will be 1, 2, 3. So this you can call it as 2-keto or alpha-keto acid. Okay, so this is how the pyruvate is produced. So these are the 10 steps, and this slide sort of tries to provide us an overall view in terms of summarizing glycolysis. So if you look at it, so the first step and the third step, and then the cleavage into the trioses. So these are the preparative phase. Okay. During which the compound, the reactants are energized and activated for the energy yielding phase. and the respective enzymes are listed here. We have already gone through it. This is just to give you a snapshot of the, the reactants and products and the enzymes, whether the reaction is reversible or irreversible. See, look at the first one. This is an irreversible reaction. Third one, it's irreversible reaction. So this catabolic reaction, therefore, is favorable only in the degradative direction. Okay. And the anabolic thing, therefore, will have to have an alternative way of converting fructose 1,6-bisphosphate to fructose 6-phosphate, as well as glucose 6-phosphate to glucose. It is not going to be the same enzyme. Like, aldolase is not going to, third, phosphofructokinase is not going to do this reversible reaction. Similarly, hexokinase is not going to do this reversible reaction. So this is how anabolism and catabolism are separately controlled, and therefore they don't run into a futile cycle. So remember, these concepts we learned at the beginning. And this is the payoff phase, meaning energy yielding phase, where we see this aldehyde group oxidation, that oxidation energy is used to reduce NAD to NADH, as well as to generate this high energy compound by making a mixed anhydride structure. And that during hydrolysis, you generate producing 2 ATP. And so that is one payoff. Then you have the second payoff at this phosphoenol pyruvate to pyruvate. And the respective enzymes are shown here again. So here, if you see, there are 2 steps again that are irreversible, like step 7 as well as step 10. So this is why it is not, pyruvate is not automatically going to become glucose. Like, for example, if you take alanine, amino acid, and remove amino group from it, it will become pyruvate. And that pyruvate, so if you are trying to make glucose from alanine, the first step is to get the amino group out, and that usually happens by a reaction called transamination. We will learn that in detail later. And that pyruvate is not readily going to become glucose. It needs enzymes to bypass this catabolic direction of glycolysis. All right, so this sort of summarizes the various molecules from which you get to glycolytic intermediate. Okay, so all this will help you as a general knowledge as well. For example, people don't readily understand the difference between eating fatty food versus carbohydrates, that is sugar-rich food. They don't realize they are all one and the same. Okay, and that is primarily because they all get converted to the same intermediates. Remember, catabolism is convergent. That also we learned initially. So here you see the lactose coming from milk. It's a disaccharide hydrolyzed, and you get glucose, and the other galactose gets isomerized to glucose again, and you know how glucose can enter into glycolysis, glucose 6-phosphate. Trachalose, this is usually from fungi. There again, it's a disaccharide. Hydrolyze the glycosidic bond and isomerize the other one into glucose, then that glucose can enter. So lactose can get to glycolysis, trachalose can get. Similarly, sugar that you eat left and right in terms of confectionery, chocolates and other sweets, and the sugar you add to milk, coffee, tea, etc. That again is a disaccharide. When you hydrolyze and isomerize the fructose into glucose, then you can get this. Or that fructose can get phosphorylated, and then it can straight get into fructose 6-phosphate and get into the glycolytic reaction. Okay. And there is one more, fructose 6-phosphate, and then that directly becomes fructose 1,6. So this aldolase can cleave into glyceraldehyde and dihydroxyacetone phosphate, and this isomerizes and then enters again into glycolysis, depending on which enzyme is present. Hexokinase reaction, fructokinase reaction. And so the other one, galactose, if it doesn't isomerize into glucose, then it can actually go via this route and then enter here. And another sugar, mannose 6 carbon, that also can enter. So like this, multiple sources of carbohydrates, they can all finally get converted into intermediates of glycolysis. And these steps are what we call as the feeder pathways of glycolysis. So now we are going to look at what actually happens to pyruvate. Okay, so from glucose to pyruvate, we have some oxidation. So an easy way of finding out the oxidation state of all carbons in a molecule is simply look at the hydrogen-carbon ratio. So if you look at it here, compared to glucose, here you have less hydrogen, meaning along with the proton, electrons are also lost, meaning it's oxidized. So pyruvate is oxidized compared to glucose. But it is still less oxidized compared to carbohydrate, meaning we can still lose electrons from it and generate more energy. So we are going to start with this pyruvate and then look at what are the things that can happen to pyruvate. One of them is immediate reduction to lactate, okay, lactic acid. This is what happens in your muscle cells when you go for a fast running, like sprint, a short sprint. So that time the oxygen transported from lungs to the muscle cells is not, the rate of transfer does not provide sufficient oxygen for complete oxidation to carbon dioxide. So as a result, the muscle that is requiring energy in a higher rate because you are running fast generates by quickly partially oxidizing glucose in the absence of oxygen into lactic acid. So the glucose entering into glycolysis comes to pyruvate, and as end of pyruvate, end of glycolysis, pyruvate gets reduced to lactic acid. And this reduction uses the NADH and in that process NADH gets oxidized. Okay, so remember at the earlier step, that is glyceraldehyde 3-phosphate dehydrogenation to produce 1,3-phosphoglycerate, there we generated NADH. And if this NADH is not produced through oxidative phosphorylation or other means, then glycolysis cannot continue because the cell will run out of oxidized form of NAD. And this lactate dehydrogenase step regenerates that. So that is summarized here. So glucose to pyruvate, you end up reducing NAD. So this is what is happening at the glyceraldehyde 3-phosphate dehydrogenase step. And that reduced one, the 4 electrons that are here, if it goes to electron transport chain, that is when oxygen is available in mitochondria, this can be used to generate proton gradient. But in the absence of oxygen, for continuous breakdown of glucose, because glucose to pyruvate, you will still get 2 ATP, and that 2 ATP could be useful to provide the energy for you to run. And that ATP to generate, you need continuous availability of this NAD. And that is generated by converting pyruvate to lactate. So in this process, NAD is consumed, and therefore this further oxidizing replenishes NAD required for the glyceraldehyde 3-phosphate dehydrogenase step. And in that sense, this becomes very crucial for anaerobic production of ATP from glucose. This is what our muscle cells do. But this can't go continuously. You could have, drunk a lot of sugary syrup and you could be running and you don't care inefficient production of ATP because you can produce a lot of glucose, and therefore even at 2 ATP per glucose, you can really make a lot of ATP. But the problem is the lactic acid is going to reduce the pH, and due to that you will have problems. So as a result of acidification of muscle cells, you can't really run too fast for long. Maximum 1 minute you can sustain the very high speed running. And beyond that, you'll have muscle cramps due to pH reduction. And this lactic acid, if you count the carbon-hydrogen ratio, is the same as glucose. Okay, so the oxidation state is not changed. The energy is still conserved here, except that you gain 2 ATP molecules per glucose. So this is one fate of pyruvate. That is, pyruvate can be reduced by lactate dehydrogenase to make lactic acid. And this lactic acid during the recovery period is transported back via blood to liver, where the liver will use this lactate to make glucose again. And that glucose can go back to muscle to feed into glycolysis to come all the way to pyruvate. In the process, you generate 2 ATP molecules. So that is the relationship between muscle and liver during that kind of active exercise. So this sort of a process where you do not require or you do not use oxygen, but derive energy out of carbohydrates, and that is the definition of fermentation. Okay, so fermentation is abstraction of energy from carbohydrates without using oxygen. So that is the biochemical definition of fermentation. So in yeast, on the other hand, unlike our muscle cells, the yeast has an interesting additional enzyme which we do not have, is the pyruvate decarboxylase. This pyruvate decarboxylase will remove this carbon dioxide from pyruvate, produce acetaldehyde, which in the presence of alcohol dehydrogenase. Remember this enzyme we have encountered as an example of competitive inhibition while studying enzyme inhibitions. Okay. Using alcohol dehydrogenase, just like lactic acid, here again the same. The reduced NADH is oxidized, and that is used to reduce acetaldehyde to ethanol. Again, ethanol, like lactic acid, has the same hydrogen-carbon ratio as glucose. So therefore, the oxidation state of the molecule is not changed, but then you have gained that 2 ATP at the phosphoenol pyruvate to pyruvate step. That 2 ATP is still net gain. So this is alcohol fermentation. An interesting aspect of this 2 enzyme step is the production of carbon dioxide. And that is why beer, champagne, etc. Have bubbling property. That is coming from this carbon dioxide produced during alcohol fermentation by yeast. And this carbon dioxide is the reason for the natural carbonation of beer and champagne. Okay. And the other very important point I want to draw your attention to, our third B complex vitamin enters into the picture, which is thiamine. Okay, so we have learned nicotinamide adenine dinucleotide, where the vitamin B complex vitamin niacin we encountered. Okay, niacin is part of NAD, and we know NAD is an important intermediate electron carrier. And then we saw another one, riboflavin, that's the vitamin B, which is part of the FMN and FAD, flavin mononucleotide and flavin adenine dinucleotide. And this is the third vitamin, thiamine. Okay, so thiamine is important cofactor for enzymes involved in decarboxylation, as you see in this example, or where acetaldehyde group needs to be temporarily transferred from one substrate to another one. So these are the 2 steps in which this thiamine becomes important. So therefore, we will spend a couple of minutes on TPP, and then we will move forward with pyruvate. So far we have seen two things about pyruvate. One, pyruvate can become lactic acid and thereby regenerate the oxidized NAD required for earlier step in glycolysis. Second, in yeast, due to the action of pyruvate decarboxylase, pyruvate can become ethanol via acetaldehyde. So this is the alcohol fermentation, where glucose is fermented to ethanol in the absence of oxygen. Okay, so now to look at the beauty of thiamine pyrophosphate structure. So what we need to do is we need to focus on this ring. So this is the thiazolium ring. So this carbon 2 is acidic, which due to the electron deficient structure of this ring, and that readily dissociates a proton and forms a carbanion ion. And that carbanion ion, as we have learned very early, will very readily react with the carbonyl carbon. Okay, because carbonyl carbon is positively charged and then carbanion will be negatively charged. So, and which results in, that usually results from a cleavage. When this carbanion reacts with a carbonyl carbon, you have a carbon-carbon bond cleavage in the molecule in which the carbonyl is present, carbonyl group is present. And that leads to a carbanion generation, which is highly reactive and unstable. And that is usually stabilized by the formation of this acetaldehyde structure here, where that additional electron is delocalized through resonance in this ring structure. And this is a common recurrent theme in biochemistry, like where you have a ring structure which is electron deficient, which can temporarily delocalize electrons through resonance formation among these double bonded ring structure. Okay, and that theme will reoccur again and again, and such ring structures we call as electron sink. Okay, so I'm sure you may have learned this in chemistry somewhere, but I just want to refresh your memory. So these are called electron sinks. Okay, this kind of a structure. That's an electron sink. So we'll see this in an actual reaction. We'll look at the pyruvate decarboxylase reaction mechanism, and there we'll get a clear idea of this. So this is a very crucial thing. So thiamine pyrophosphate, here we are seeing with pyruvate decarboxylase as example, and I said this enzyme is present in yeast and not present in us, and therefore I don't care about, don't think that way, or this vitamin may not be important for us. There are numerous decarboxylation steps in our body, as well as there are many situations where acetaldehyde group transfer is important. Due to this, TPP is extremely important. Okay, so you will see a reaction later, not in today's class, where we are going to proceed from pyruvate to the oxidative phosphorylation step, where the oxidative phosphorylation and pyruvate is linked by a cyclical metabolic pathway called the citric acid cycle or Krebs cycle. At the junction between glycolysis and the TCA cycle, or tricarboxylic acid, because you have 3 carboxyls in one molecule there, and in that between the two is pyruvate to acetyl-CoA formation, and there you need TPP again. So it is extremely crucial, and due to that, thiamine deficiency can be fatal. Okay, you can ultimately lead to death. So thiamine deficiency causes beriberi. Remember, niacin is pellagra. So We saw rickets with respect to vitamin D. That's a lipid soluble vitamin. So beriberi leads to is characterized by fluid accumulation, pain, paralysis, and ultimately death. So this is an important vitamin. So usually we don't suffer vitamin B deficiency primarily because most of the food that we eat, in some or the other, you get vitamins, the B complex vitamins. These are water soluble vitamins. They are readily excreted, and as a result, hypervitamin based diseases do not happen. That's called hypervitaminosis. That is not associated with B complex. And B complex vitamins are available from variety of plant sourced food, and that's one of the reasons why you need to eat a variety of vegetables in your diet. At some time or the other. So having variety in the food is one of the reasons for to fulfill the requirements of dietary requirements of all these vitamins. So normally you do not run deficiency of these vitamins, but in malnutrition conditions that can happen, and that's how we are aware of a disease like beriberi. So in India, malnutrition is still common. So therefore, vitamin deficiency is still a problem. So I can't undermine the importance of this. Okay, let us get to the reaction mechanism of pyruvate decarboxylase. So in school, you may have memorized vitamins are important. So now you are actually seeing where the vitamins actually function. So this is the thiazolium ring. So where this is little acidic carbon which can readily dissociate this proton and become carbanion. And we have already learned at the very beginning, even before we got into metabolism, that these carbanions can readily attack a carbonyl carbon because this is an electrophile and this is a nucleophile. Okay. And that leads to the formation of this intermediate. So here you have this. See, this is the ethyl group, 2 carbons, and once you remove this by decarboxylation, so this will be an ethyl group. So this is a hydroxyethyl. Okay, when you have another hydrogen for this, as you see here. So because this can readily delocalize electrons, that favors this decarboxylation, and then you get this structure. And this structure, because of the delocalization of electrons, this is the resonance stabilization between the two forms. So you have either this double bond or the carbanion, and both of this can readily be stabilized by delocalization with among these atoms, among these bonds. Okay, and then protonation leads to the formation of this hydroxyethyl structure. So this is how acetaldehyde is carried, and therefore this hydroxyethyl structure is normally called active acetaldehyde group. Okay, so this is actually activated form which can readily react, meaning here readily donate the acetaldehyde to another substrate. So that is the kind of reactions in which TPP becomes important. So when we learned about the type of reactions, first we learned about oxidation reduction reaction. Second, we talked about carbon-carbon bond formation or breakage. There we saw the importance of carbonyl carbon, and we learned about aldol condensation, glycine condensation, and the third was alpha-keto acid decarboxylation, and that is the example here you are seeing. Okay, so here carbanion, and you have the carbonyl carbon, the positively positive carbon, and that results in decarboxylation of this alpha-keto acids. This is the alpha carbon. So we don't count this. In IUPAC, this is 1 and 2. So pyruvate would be 2-keto acid or alpha-keto acid. So the alpha-keto acid carbonyl group is the one that favors the decarboxylation of such acids. Okay, here you see an example of that. And then, so again, the rearrangement of electrons here generates acetaldehyde and produces TPP. That by taking a proton, losing a proton into the medium, generates this active carbon ion version. Okay, so these are the 5 steps of the TPP based decarboxylation of pyruvate decarboxylation. So this is an important enzyme reaction mechanism. Make sure you go through this multiple times. The main point is this carbon ion and carbonyl carbon reaction. Okay, so far we have seen glucose to pyruvate to lactic acid as well as pyruvate to ethanol. Okay, so a third fate of pyruvate happens when you have plenty of oxygen available, which is the normal situation in all our cells. There the pyruvate gets oxidized to carbon dioxide. Remember, in terms of the oxidation states of carbon dioxide, this is the most oxidized stage. So this happens when oxygen is plenty, and that is aerobic conditions, during which pyruvate gets converted to acetyl-CoA, which enters into a cyclical biochemical pathway called citric acid cycle, because one of the intermediates of the pathway is citric acid. Citric acid has 3 carboxyl groups, and therefore it is also called tricarboxylic acid cycle. Or in memory of or in honor of the scientist who discovered this pathway, it is called Krebs cycle. Okay, Krebs cycle. So Hans Krebs is the scientist who discovered this pathway. So our next goal will be that. So we will learn about the TCA cycle. But before that, we have to do a few small, finishing up of the glycolysis. One of them is glycolysis regulation itself. Glycolysis is an interesting pathway that is the central, as I told you at the beginning, to metabolism in all organisms. It's subject to regulation. And one of the main point is, in the absence of oxygen, glycolysis is the main source of energy. Okay. So, for example, if you take a cancer tissue, a tumor that is growing, initially when the tumor starts to form, as it enlarges, it will not have induced blood vessels to supply blood to the inner parts of the tumor. Due to that, it will not have plenty of oxygen available. So the oxygen in our tissues is primarily from the blood, and the oxygen in the blood is from the lungs. Okay, so lungs is where from the atmospheric air you have the oxygen loaded onto hemoglobin, and hemoglobin in the blood takes the oxygen to tissues. Now, if a tissue doesn't have blood vessels nicely supplied, then it is not going to have oxygen. So, to draw enough oxygen and other nutrients, cancer tissues, the tumors, induce blood vessel formation, a process called angiogenesis. But when they are still in the process of inducing and the angiogenesis and the blood vessels have not yet formed, the middle of the tumor will not have enough oxygen supply. So due to that, they rely on glycolysis to get enough energy for the cell proliferation. And due to this, tumors are highly active in glycolysis. And this process was originally identified that tumor cells have higher rate of glycolysis than other tissues or the normal tissues was identified by a scientist by name Otto Warburg. Okay, and due to that it is called Warburg effect. Okay, so this tumor having high glycolysis. And even before him, Louis Pasteur, growing yeast and other microorganisms, realized in the absence of oxygen in anaerobic conditions, glucose breakdown was happening at a higher rate, and that is because glycolysis is the primary way of getting the required ATP molecules, which means you need to break down a lot of glucose. And due to that, glucose uptake by the microorganisms in the absence of oxygen was high, and that is called Pasteur effect. Okay, so Pasteur effect is higher glycolysis in microorganisms in the absence of oxygen, that is anaerobic conditions. So this was observed by Pasteur, and that's called Pasteur effect. Then Warburg discovered the same thing in tumor, and that we call as Warburg effect. Okay, so remember those things. That's about the regulation of glycolysis. So next what we are going to do is we are going to look at the reverse of the whole thing to make glucose. So why would you make glucose? You know, if glucose is not directly currency of energy, and I needed to oxidize glucose to make ATP, and we saw through the feeder pathways, many other molecules can actually be converted to intermediates of the glycolysis, and we can get energy from them as well. If that is the case, why would I ever need to make glucose? That is because other molecules don't readily cross the blood-brain barrier, and brain depends on availability of glucose for all its energy needs. And among the organs in our body, the one that requires the maximum energy is our brain. So that is why if the blood glucose level goes below a certain level, you get into coma. Okay, and that is called hypoglycemia. So you can't have hypoglycemia, you will immediately get into coma. So you would think too much glucose is always bad because that is what is diabetes is all about. But the opposite is also, the opposite is also true. That is primarily because of the way our brain uses, depends on this. So hypoglycemia again is a serious condition. Like this is lowering of glucose in blood, and due to that, brain will not have sufficient energy, and due to that its activity will go down, and then you will get into coma. To avoid that, glucose production is important. Maintenance of the normal levels of glucose, like roughly about 70 milligrams per 100 ml of blood, is really crucial. If it goes below 40, you will go into coma. So therefore, how do we make glucose? From other things. Suppose glucose is not readily available, and that's the reason you will not have glucose in the blood. So there are a variety of ways. So I'll complete that and then stop. So lactate is one source. Lactate produced in the muscle comes to liver, and liver converts lactate to pyruvate by the reversal of the lactate dehydrogenase step that we saw already. And this pyruvate can get into citric acid cycle and then go back to glucose. So pyruvate citric acid connection I will tell you in the next class. So this is one route. The other one is triacyl glycerols. The fatty acids in acyl form ester bond with the glycerol. So they can be hydrolyzed, and this glycerol can become glyceraldehyde 3-phosphate and enter into glycolysis and go back. And free fatty acids also can go, that let us not worry for now. Then plants and some microbes like cyanobacteria, they can fix carbon dioxide using sun's energy and produce 3-phosphoglycerate, which can also get into glycolytic intermediate and go back to make glucose, which can go into making all of this. Like an important thing in hypoglycemia is blood glucose. Suppose you are eating only meat and never eat any carbohydrate food, then you are depending on amino acids for producing blood glucose. Okay, so there, like for example, alanine transamination can generate pyruvate, and that can go into forming blood glucose. Excess of glucose produced even that way can become glycogen in liver. And glycoproteins, we know what they are and what's their use. Disaccharides like lactic acid, lactose in mammary gland, or sucrose, maltose, all of that. Other monosaccharides, and in plants, of course, starch and sucrose. So this sort of reversal is what is gluconeogenesis. Okay. So in the next class, we will look at the, I'll just briefly show you. So this is the glycolysis that we already saw, glucose to pyruvate, which is marked in the pink color here. And I told you the reversal cannot happen by identical enzymes. So what are the steps? The bubble-like structures. That we will focus in detailed debate tomorrow. Okay, so I'll stop here.